Capillaries are the smallest and the most numerous blood vessels that are present in our body, and they form a vast network that connect the arteries, the vessels which carry blood away from the heart, to the veins, the vessels which carry blood towards the heart. So there are three types of capillaries: continuous capillaries, fenestrated capillaries, and sinusoidal capillaries. So in this video we are going to discuss the differences between these. So let's cut this table into two halves so that it is easily visible. So let's discuss the first feature, that is endothelial cells. In continuous capillaries, these cells are tightly joined with each other and they form a continuous lining. You can see in this diagram. In fenestrated capillaries, these cells have pores, which are also known as fenestrae. In sinusoidal capillaries, they have large gaps between these cells, so they don't form a continuous lining. That's why they are also known as discontinuous capillaries. Okay, the next feature is cell junctions. In continuous capillaries, tight junctions are present. In fenestrated capillaries, tight junctions are also present. Whereas in sinusoidal capillaries, there are no tight junctions. The junctions are absent or incomplete. The next feature is basement membrane. It is continuous and complete both in continuous capillaries and fenestrated capillaries, whereas it's discontinuous or absent in sinusoidal capillaries. The next feature is pores or fenestra. So in continuous capillaries, they are not present, whereas in fenestrated capillaries, yes, but they are covered by diaphragm. Whereas in sinusoidal capillaries, they have large gaps and no diaphragms are present there. So now next move to the second part of this table. So now let's talk about the permeability. The continuous capillaries, they are the least permeable. The fenestrated capillaries, they are moderately permeable, whereas the sinusoidal capillaries, they are the most permeable. Okay, now size. Continuous capillaries, they have the smallest diameter, that is between 5 to approximately 10 micron. The size of fenestrated capillaries is slightly larger than continuous capillaries, whereas sinusoidal capillaries, they are the largest. They have the largest diameter, that is between approximately 30 to 40 microns. They have irregular shape. Okay, now key location. The continuous capillaries, they are present in skin, muscles, lungs, and CNS. Okay, fenestrated capillaries, they are present in kidneys, intestines, and endocrine glands. Look, you can see that their location is where reabsorption or absorption or some exchange is taking place rapidly. Okay, now sinusoidal capillaries, they are present in liver, spleen, bone marrow, and lymph nodes, and their location is very important. Okay, now let's move to the primary function. Continuous capillaries, their function is selective exchange and barrier protection. Sinusoidal capillaries, their function is blood cell passage, protein exchange, and immune surveillance. Whereas our fenestrated capillaries, as I told you earlier, they are involved in rapid fluid and filter exchange. So if you like this video, click on the like button. Thank you so much.